Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Dr. Josefina Nilsson of Vironova.
Thank you, Leah, very much for the presentation. And thank you all for signing in for this webinar. Today we will be focusing on characterization on drug and gene delivery platforms, and then most specifically structural characterization that can give you important insight into your quality of your development and manufacturing processes, which can of course then also be connected to your encapsulated system biological activity, biodistribution, and toxicity. You can, of course, pack different kind of active substances into different encapsulation systems. And depending on what active substance you are using and where you want it to be delivered, you will choose different systems. However, independently on what system you are choosing, there are specific structural characteristics that are common and can give you important insights. And this might be like the morphology, the integrity, and also the filled versus empty particles. There are, of course, also unique parameters specific to specific systems. For instance, if you're working with lipid-based systems, it might be that you would like to look into the size distribution, the level of lamellarity, as well as the thickness of the lipid bilayer. However, if you're working with viral vectors, it might be that the size distribution is not that important, and that it's more the aggregation of your particles and purity of your samples that have with your quality to do. When it comes to the regulation requirements, for instance, when it comes to liposomes, there are guidelines from the FDA, and they are listing a lot of different parameters that they think it's good to look into. And I show you a few of them here, and I marked a few of them with a star, where we can actually help you looking into these by using electron microscopy and image analysis. And the beauty with this is that by one and a single analysis, you can get insight of all of these parameters. However, when it comes to the viral vectors, I at least have not been able to find a similar document to the FDA guidelines for liposomes. However, if we collect the information and questions that we get from our clients, I think that these parameters are few that are important, like the morphology, integrity, and purity of your particles. So during today's webinar, I will be focusing on the analysis using electron microscopy and image analysis, specifically for gene delivery systems using viral vectors and also other kind of drug delivery systems using liposomes. I will show you a little bit about how we work here at Vironova, and also case studies of the work that we do routinely here. So, after we have done our sample preparation and image acquisition using electron microscopy, we use our in-house developed software to do the image analysis. And the beauty with this is that we move electron microscopy from being thought of as the subjective and quite time-consuming analysis method to a method which actually gives you objective, repeatable, and quantitative information. So after we have done the image acquisition, we load up all our images into this system. And we let it automatically detect all the particles within the images, and we can let it run through a number of images or actually stop when we have reached statistical significance to the question of interest. After that, we can use the same software to classify different particles within the images. These kind of classes might be empty versus filled particles, unilaminar versus multilaminar, different shapes of particles, or different sizes. When the analysis is done, we generate a report, and this report will then contain information that we have added into the system regarding the sample preparation, image acquisition. You will, of course, also get representative images of the sample, histogram, if it's so that we have done size distribution, for instance, and all the statistical data. You will also see, like this bar chart, if we have done some kind of classification, like filled versus empty particles. So this is more or less the workflow that we are doing here. So due to the short time of this webinar, let me jump into some case studies where we start with viral vectors. So in this first case study, it was done on adenoviruses, and we looked into samples with different formulations. And in the image on your left-hand side, you can actually see particles, adenovirus particles, which look very nice. It's a good integrity, nice morphology, and you can see that these particles are shown as single units. However, when the client made a change in formulation, you can see the image, a representative image on the right-hand side. You can see that the particles actually start to break down and also aggregate. So independently of what kind of particles you're working with, you don't want your particles to end up like this. Another case study where we looked into the purification step. This time it was another viral vector. It was the AAV, the adeno-associated virus. On the left-hand side, you can actually see it's a low magnification image. You can see the particles in the background, and these big black dots are actually aggregates, as you can see here on the right-hand side. Except from the aggregate, you can actually see in the background here, it's quite noisy. All these small white dots are some kind of impurities, probably protein-based. You have your particles looking rather nice, but you also have some kind of structure here in the background, which is actually proteasomes shown at different angles. So here again, we looked at purification steps before and after certain filtration. And often you think that you remove aggregates by filtration, but here we actually increase the aggregation due to stress of the sample. So these two first case studies shows quite normal image analysis or EM analysis that you would think of. When adding our electron microscopy or image analysis tool to this, we add on a layer of more objective, repeatable, and quantitative data. Again, this is the AAV vector. You see the particles here, and you also have the proteasomes at different angles. So we have run through a couple of images using our software. They have detect the particles and classify them due to size. So here we have the peak for the AAV and the proteasome, and compare this data to a DLS run where the DLS was not able to differentiate between the two peaks. Last but not least, is a very important study when it comes to AAV and other viral vectors, and that is, of course, the filled versus empty analysis. Here again is picture on the left-hand side of the AAV vector. We are using cryo-electron microscopy, so we are not depending on any stain penetrating the virus particles of the empty one. So we take the images and we let the software run detection and classification, and you can see that the software easily can differentiate between the empty particles shown in blue compared to the filled particles shown in red. So these are 4 case studies that we often do for biovectors. So move on now to another vector system, the lipid-based system. Here we show liposomes packed with dock seal. You have the dock seal here as the bar structure in the middle. This was a stability study that we ran on the liposomes. And here we compared the size with the shape of the particles to find out the optimal storage conditions for these particles to make sure that the size and the shape remained. And at the same time, on the same analysis, we can also then use the software to differentiate between the filled and empty particles. Here again, the dock seal in the middle. And another classification that we can do using our software is the lamellarity analysis. Here we have empty liposomes, and you can clearly see or differentiate between the unilamellar and the multilamellar. And depending on what kind of drug you want to pack in your system and where it is, it might be that you would like to end up with either these or these. And due to the high resolution in the images, you can very easily see the lipid bilayer, and you can actually differentiate or see the phospholipid heads of the lipid. And due to that, that is also something that we can then automatically measure with our software. So here we can then report the lipid bilayer thickness, which is often required from the authorities. So these were actually the case studies that I wanted to show you, but I hope that I've been able to show you that you can get a lot of information from electron microscopy and image analysis. So by using our EM services, you will directly get in touch with our experts in electron microscopy and image analysis. Normally, you will receive the results within 10 working days from the day that we have or received your sample. You will get, of course, all the acquired images, the images with detected particles, and report containing representative images with a quantitative analysis, statistics, and also a concluding summary from our experts. So we think, of course, that structural characterization is of utmost importance when it comes to having an effective and high-quality R&D and manufacturing process of drug delivery systems. Using electron microscopy and image analysis will give you objective, repeatable, and quantitative structural information, and you will get a good and cost-efficient way where you can have all the information, structural information needed for one and the same analysis. So I'm afraid that I've run out of time, and if you have any questions, please go ahead and write them in the chat window, or you can contact me afterwards and I will try to answer all the questions you have. With me here today to answer the questions, I have two of my colleagues, Gustav Kihlberg, which is expert in image analysis, and Mathieu, which is one of our best electron microscopists. Thank you very much.
Thank you, Josefina. So to start with, we have an audience member who noted the nicely stained samples and wondered, were they PTA stained, or can you talk about that?
Yeah, it was urinal stain, urinal acetate.
Okay. And do you prepare GMP reports and analysis as part of services?
We have a quality system that we are working according to, yes.
Okay. And how do you ensure that the liposomes in the cryo-EM images are representative?
So here's Mathieu speaking. So we always try to make the image data set as representative as possible by exploring the whole area of the grid. So usually we look at areas with different height thickness when it comes to cryo-EM, and just in order to get sure that what we provide to the customer is representative of what was in the sample.
And how can you ensure that morphology of the liposomes are representative and not affected by the preparation procedure?
So when we work with cryo-electron microscopy, we are actually freezing the sample in liquid ethane. And it's a really fast freezing, so it's called the flash freezing, that goes from room temperature to cryogenic temperatures within milliseconds. And so that we end up with ice in a vitreous state to which we can see through, so it's not crystalline ice. And this fast freezing provides us with a good overview of the sample and makes sure that we don't affect the sample morphology or the sample integrity. So the fast freezing is the key to be sure that we don't affect the liposome integrity as well.
Okay. And how many particles do you measure?
So that depends on how many we have on grid, but usually we try to make sure that we have something which is representative, and our threshold is of 1,500 particles. So we make sure that if the concentration is sufficient, we try to analyze at least 1,500 particles to be sure that we get a representative population with some statistically relevant data.
And can you measure PEG layers of liposomes using any of your electron microscopy techniques?
So we cannot see the PEG layer because they are not dense enough to be seen by electron microscopy. The PEG layer are composed mainly or exclusively of oxygen, hydrogen, and carbon, and this doesn't give enough contrast with respect to the support. So the support is made of carbon, and the PEG molecules are also made mainly of carbon, which doesn't provide enough contrast, and unfortunately, no, this is something that we cannot provide you.
And how do you discriminate between filled and empty vial particle vectors, for example, packaging of AAV particles?
So when it comes to AAV, we observe them by cryo-EM, and then we discriminate between empty and filled by doing a so-called radial density profile. So basically, we look at the particle, we look at the profile of the particle, and already by eye you can see that the empty ones will appear as an open circle, while the filled one will appear as a disk. And then by automating an analysis on this particle with the software, by looking at the radius or the radial of the particles, you can see that the profile will differ depending on if you have a disk or a circle. And then this is what Josefina has been presenting when we saw that there was this cluster that was separated, and this is how we end up. So we look at the whole AAV population, we check the profile, then we plot the profile on the plot, and we obtain clusters, and usually if the definition is good enough, the clusters are well separated, and then we can discriminate between empty and full, and this is how we do it actually.
And is it possible to accurately gauge particle or liposome concentration by your software? And if so, has it been compared to alternate techniques?
are you talking about assessing the concentration of liposomes? Is it right?
Yes, accurately
That's
gauge particle or liposome. Yes.
So we do not do any concentration, or we don't quantify liposomes in terms of concentration because the EM is not suitable at all to get any quantitative data when it comes to number or concentration of particles. So when Josefina was talking about quantitation, it was relative quantitative analysis. So when we look, for example, at field versus empty, we get some numbers, and it's relative field versus empty. But if you want to have a concentration, EM is not a suitable method for this.
Okay, well, thank you so much for answering the questions. Thank you, audience, for asking questions. Thank you, Josefina, for your presentation.
Thank you very much.
Thank you. And we'll be posting the recorded version on our website shortly. Thank you.
It came in a really long question just before